All right, let's start. This is week 5. I'm Soumyadeep. Thank you for joining this session. This is a NPTEL PMRF session for the course of Tissue Engineering by Professor Vignesh. So it is now we are in week 5. So I hope you guys are learning as much as possible, and you are going through the lectures and having a good examination experience. So how are your examinations going? Let me just have a poll here. How your examinations are going right now? So right. Right. So you are going through all the course contents like the whatever the slides were there, and as well as the supplementary material that were there. I hope you are doing that. So it will be like helping you to learn all the things. Okay, that that the message is being trying to convey. Right? Okay. Cool. Now, so this week we have learned about various cell sources. We have had a previous discussion about the autologous and xenogenous cell sources, various different type of cell sources. When we were studying the hydrogel sections. Now this week we particularly tried to address the various cell sources and mostly focusing on the stem cells. Okay, stem cells for various kinds of stem cells like pluripotent, totipotent, multipotent stem cells. What are the sites they are harvested from? What are the stem cell niches that are very important for its particular tissue engineering applications, and how its biology changes. Okay. Along with that, we have studied about cell isolation techniques, cell harvesting, cell retrieval methods, cell identification techniques, whatever the density gradient centrifugation, FACS, MAX, and other techniques that were discussed. Okay, as well as tissue dynamics, we also studied the tissue behavior changes. Previously, we have been studying about the cellular behavior from the tissue engineering perspective. Now, this week we have studied about the tissue dynamics part, like how the tissue behavior changes and how the tissue behavior impacts the tissue engineering perspective. Okay, then we have focused on the cell differentiation factor, uh, mostly concerned about the the hematopoiesis steps, as well as factors regulating the differentiation, de-differentiation behavior, induced pluripotent stem cells. These things we have studied this week. Okay. Along with that, I wanted to start off with a very basic recap, like what we have been studying in this PMRF sessions. So I hope you remember these mechanisms we have taught, like glucose sensing mechanism, like both natural and synthetic, classical mechanism, right? Glucose sensing mechanisms we have studied. So how the ionic imbalance, the osmotic imbalance creates the swelling of the hydrogen, as well as the increase in insulin release, cargo release, and the PBA version of the synthetic version, the phenyl boronic acid based synthetic polymers, which can be used as a glucose sensing system. We have studied about it, right? Then we also have studied about the biomimetic anisotropic hydrogels, why the anisotropy is important, how the induction of anisotropy helps in the tissue engineering perspective, how the anisotropy is created in the hydrogel platform by introducing another phase to the hydrogel, not only just the hydrogel phase, another phase by different mechanisms. You can put the different phase separately, or you can make it in situ by another fabrication techniques like mechanical stitching, or as it is being represented in B, right? These mechanisms and anisotropy and importance of biomedical anisotropy and biomimetic anisotropy we have studied, right? Next, we have also studied about the electrospinning process. What are the electrospinning techniques available? How the spinnerets and collecting surfaces, that is the stage where the collection is happening of the nanofiber. This changes, how it is changes. We have studied about it, and what is this called? This B, you can see this, right? This B, what is this called? When electrospinning is occurring with the creation of very high electric potential, what structure forms first at the tip of the needle? What do we call it? We call it a, try to remember. a Taylor cone, right? We call it a Taylor cone. So the first Taylor cone forms, then the polymer whips out and collects as in the stage. There are various kinds of stage also we have studied about, like not only just a static stage, we can have a uh, rolling surface also, as well as have the various other kinds of surfaces depending upon the application in our hand. Okay, that we have studied. Then we also have studied about shear thinning, self-healing hydrogels, shear thinning property, how shear thinning mechanism works, the polymers reagent in polymer rearranges when the shear is there, right? As the shear is applied, the polymer molecules rearranges. Therefore, this thinning property comes. Where it is applied for injectable hydrogel systems or specifically in 3D bioprinting. The bio inks, remember bio inks that we prepare there, the shear thinning property is very important, right? So shear thinning we also have studied. Next, we also have studied about the scanning tunneling microscopy. You remember, right? Scanning tunneling microscopy, where very small gaps are there so that the tunneling current occurs, so that we can get the atomic level information about the surface. And what are these tips are made of? Tungsten, platinum, iridium. What are the challenges? Like conducting material is only needed, vibrational interference and contamination on the surface. This kind of problems are there with the scanning tunneling microscope, but there is a very high resolution of the surface information that we can get. Other techniques, what is the modification from the scanning after the STM? What is the technique that came to minimize these challenges? What are the modifications? What is the technique that came upon after this after STM to remove these challenges and we can get a better surface topography? It is atomic force microscopy, right? AFM. AFMs we have got three modes. What are the three modes in AFM? Atomic force microscopy. What are the three modes that we have? Three modes of operation for AFM. So scanning tunneling microscope, then came the AFM to get the topography. So what are the three methods? What are the three things that we can three modes that we can perform the AFM? Tapping mode, contact mode, non-contact mode, right? So if you remember, right? You try to remember, try to recall. That's why I kept this recap session that like it's this almost a 50% of course is done. Now just try to remember whatever we have learned till now, so that it will be beneficial when the final examination dates come, right? Okay. So the scanning tunneling microscopy we also have learned, right? Along with this property, stress strain behavior, how the metal and polymers behave differently, this thing we also have learned. So the neck is created, while in case of metal, the neck doesn't get elongated much. The breaking point comes much faster. But while in case of the polymers, the necking comes and all the rearrangement of the polymeric molecules happens in this necking region only. And whatever the elongation and whatever the elasticity or plasticity that is shown, it is in this necking region only. Right? We have studied about it all. Okay. So with this recap, I hope you know, time to time you try to uh, repeating all the things that we have learned, because it's a 8 week long course. So it will be helping you when the final dates come. It will be helping you to remember or the remember and write in the exam. Right? Okay. Right. So to start with, I have this image. So it is question set, like a 4 5 questions set based on this image only. Identify the type of stem cells that are being represented in this image. For example, the question 1 here, it says that choose the type of cells you find in location marked as 1 in the figure. Options are pluripotent stem cell, multipotent stem cells, totipotent stem cells, unipotent stem cells, and somatic cells. You can see this 1 here. What is the kind of? Okay, it is totipotent stem cells. Right? See, uh, when we are talking about this kind of embryonic, pre-embryonic stages of cells harvesting, then their differentiation potential is much, much higher. We will be discussing about it after the set of questions goes on. So this differentiation would be potential would be much, much higher. So this kind of cells can differentiate into any kinds of cells that are constitute that our body is constituted of. So when this kind of embryonic stage we are talking about, early embryonic stage, it is almost always totipotent stem cells. Totipotent means it can divide into almost every kind of cells, right? You don't have to remember or like mug up it. So just think about it. It is the early stage of your differentiation or developmental stage. So the cells that are produced there are of very much potential, have very much potential of developing into any kind of cells, because those are the primary cells from where your all the cells are being made of, right? So depending upon that, with this information, we know that it would be like totipotent cells, right? All right, let's see the next question. Choose the type. Sorry, I go the answer. Choose the type of cells you find in the marked as 2 in the figure. It would be pluripotent stem cells, right? Here, so see, when the primary cells like this 4 cell stage to the morula stage came in, cells are almost like having the potential of all the type of cells. After the blastocyst and inner cell line form stage goes in, the cells have almost decided, decided that yes, my direction of differentiation, the fate of my differentiation will be like in this direction, in a particular direction only, not to the level of our unipotent or multipotent cells, but here their differentiation fate is getting narrowed. Right. So as the cells are getting divided from that two cell stage to four cell stage to morula stage, and from later on, as the cell divisions are going on, their differentiation fate is also getting narrowed. Right. So the early stages of the cell differentiation, their potential to differentiate into other kinds of cells is much more higher. So whenever you find a question where you see that the stage of the development is much earlier, you always think about that too, like pluripotent or totipotent, right? So keeping these things in mind, whenever we are talking about these embryonic developmental stages, we have like first, if we get to the like fourth cell or morula stage while the cells are being taken out, that would be always totipotent. And when their invagination of this development is this stage has come up, so there will be like pluripotent. Okay, right? All right, let's go to the next question. Somatic cells shown with the marking 3 in the figure undergoes which of the following processes in order to give rise to iPSCs? Somatic cells shown with marking 3 here in the figure undergo which of the following process in order to, all right, differentiation, redifferentiation, differentiation, and transdifferentiation. Right, this question in 3, what is the process? What is going on to give rise to? Okay, all right. Let's see, there is a confusion. It is de-differentiation. Okay. Right. Yeah. Whenever a somatic cells is turned into uh, previous, like with its differentiation potential is getting higher. So as I say, like as the cells is dividing, its differentiation potential is getting narrower, right? So it is getting towards the multipotent, then unipotent, and towards the fixed kind of cells, right? So when a somatic cells, that is, it's kind of at the end of their differentiation or developmental period, right? So when the somatic cells is getting back to the position where it can get rise to iPSCs, like induced pluripotent stem cells, induced pluripotent stem cells. You can see from the term, like it is being induced to make into a pluripotent stem cells, where it will have the property to differentiate into a myriad of different kinds of cells, right? So this process would be called as de-differentiation. Differentiation is the phenomena where the cell will attain its fate towards its final fate, like towards its functional state, where it will perform that particular function of the tissue where they are in the particular stage, right? So differentiation is the phenomena. It's like you can think about it like this way. It would be like this fourth cell stage from morula. Then it will start differentiating. That the process, it will start differentiating towards the stage where it will start functioning for a particular tissue at a particular cell niche. It will have a particular function in relation with a particular tissue, which will be having a particular function in relation to a particular organ. So it is the differentiation where the cell will gain its fate, its ultimate fate. But the reverse process, where this somatic cells, a differentiated cell, will be induced by introduction of several factors, again, the factors that you have learned. So different factors induction have given them the property of differentiating into different cells. Again, so they have de-differentiated, right? So the correct option would be de-differentiation, not differentiation or re-differentiation. Okay, it is clear, right? De-differentiation and differentiation. All right. So moving on. Right. This question: induced pluripotent stem cells (iPSC) shown with marking 4 in the figure resemble which of the following cell type? Hematopoietic stem cells, embryonic stem cells, dental pulp stem cells, modular cells, and cells from iliac crest. Yes. Okay. It is embryonic stem cells. Right. So you can trace back to the previous information that I was talking about. This is the de-differentiation that has given them the potential of pluripotency. So what is the kind of cells that have this kind of potential? Embryonic stem cells, right? So the correct option would be embryonic cells. You don't have to like mug up all this information. You just understand how this cell division and how the development stage is going on, and according to that cycle, how the cell's properties are changes. Okay, changing. Right. So the next question is: choose the type of cells you find in location 5 and 6 in the figure. Pluripotent stem cells, multipotent stem cells, totipotent stem cells, and unipotent stem cells. Choose the type of cells you find in location marked as 5 and 6, here and here. Options are pluripotent, multipotent, totipotent, and unipotent. What would be the answer? What kind of cells we can find from those locations? The answer would be multipotent stem cells, right? Yeah, multipotent stem cells. What kind of cells we can get from? So these are the locations from where. What kind of cells we can get? What is the technique? We have learned in the lecture only. Try to remember. It is called bone marrow aspiration, right? We collect bone marrows, yeah, bone marrow stem cells or hematopoietic stem cells we can get from these locations, right? So this kind of cells have the potential to generate uh, different kind of cells that are uh, hematopoietic in origin, right? Like different kind of blood cells. Okay. So this kind of hematopoietic stem cells which are multipotent in nature, not pluripotent, not totipotent, not unipotent, especially not unipotent, but not pluripotent and totipotent, because they have the fate to divide into just only blood cells, right? So these are multipotent stem cells, and the process is called bone marrow aspiration, and we can collect bone marrow from these sites, right? So we are always discussing about the stem cells property of uh, self renewal as well as producing cells that have the fate to develop into a particular type of cells. So what govern this self renewal property? What are the molecular mechanisms that occurs to regulate this self renewal phenomena of stem cells? So there are several reports that suggest this WNT, that is Wnt pathway, that is also known as SHH, sonic hedgehog, and Notch. These three molecular pathway greatly regulate this self-renewal property of stem cells. So by regulating this three molecular pathway, an element of these three molecular pathways, the stem cells differentiation phenomena can be regulated. It can be either fettered to its differentiation fate, or it can be also fated to self-renewal fate, right? So, depending upon this regulation phenomena of these three pathways, that is WNT, SHH, and Notch, this self-renewal property of stem cells occurs. Now, not only just stem cell self-renewal property has a very unfortunate resemblance to the cancer's property. So, the cancer cell also has the regulatory mechanism based on these three molecular pathways. So their self-renewal, that is, they are producing these cancerous cell types of similar kinds, also gets help from these three pathways. So not only just cancer cells, also there is another term called cancer stem cells that is of much of concern, because cancer stem cells has a much greater risk to the metastasis phenomena. So when we are talking about cancer stem cells, we are talking about the kind of cells that mediate the cancer's property to metastasize, so different locations, right? It has the property to self renewal, and then it can intravasate and extravasate, that is get from the site of its origin to the site of its where it is its second home, you can call it. So these cancer stem cells also have this regulation based on these three molecular phenomena. So stem cells, cancer cells, and cancer stem cells, all these various kinds of cells are regulated by this WNT, SHH, and Notch pathways, and their self-renewal property and their division and their growth. This all these properties are regulated by this molecular pathway. Okay, so these are the molecular backbone depending upon which this stem cells self-renewal property is coming out. Okay, right? All right. Let's move on to the next question. Which of the following cells need suitable surface for attachment for their growth and proliferation? Which of the following cells need suitable surface for attachment for their growth and proliferation? Options are given as beta lymphocyte, T lymphocyte, B lymphocyte, T lymphocyte, erythrocytes, natural killer cells, osteoblast cells, and mast cells. Okay, I'm getting responses. Right, osteoblast cells. Yes. Why not the other cells? This B lymphocyte, T lymphocyte, erythrocytes. What kind of cells are those? Right, they are like blood cells, immune cells, right? So yes, right. So these osteoblast cells are the one that needs a suitable surface for adherence. right? Right. They circulate in the blood, and these osteoblast cells need a particular kind of hard surfaces. Yeah, you can remember from the what kind of surface, what kind of material that would be suitable for this osteoblasts and chondrocytes kind of cells culturing. Soft material and hard material. Depend in these two options, what weak material? Yeah, hard substrate. We need a substrate that is much harder so that they can find their suitable niche to develop into that particular type of tissue. Here we are talking about osteoblast, so it is a surface adhering cells. Okay, so not only just these various growth factors that are required for stem cells differentiation. When we're talking about stem cells differentiations, we talk about various internal and external stimuli, the growth condition, what are the growth factors that are supplied to the stem cells. So not only just these growth factors or the cocktails of both of them, there are various other mechanical stimuli that can have a significant impact in stem cells differentiation property. One of those mechanical stimuli is one of those. Okay, this external mechanical stimuli that are supplied now being like investigated worldwide. So they are investigating what kind of mechanical stimuli and what level of them and how the cells are responding, stem cells are differentiating depending upon the external stimuli and their properties. For example, you can see in a like this top left corner, this is a uniaxial stretching unit, the basic diagram of a uniaxial stretching unit. Here you can see this two material, these are the clips to hold a substrate. For example, let's say a nanofiber, or for example, say a hydrogel block. And with the help of this actuator, so this is nothing but a motor. So with the help of this actuator, this gel or the nanofiber substrate is it's uniaxially stretched. And depending upon this stretching motion, the degree of its like the strain amount as well as the frequency, various mesenchymal stem cells have shown the property of differentiation to either osteoblastic or muscle or chondrogenic. So their differentiation pattern changes depending upon this mechanical stimuli also. You know why? Because the mechanotransduction procedure, mechanotransduction pathways kick in when this kind of external mechanical stimuli are given to the cells. And one of the components of this mechanical mechanotransduction is WNT pathway, notch pathway. So these are the uh, prime regulator of this mechanotransduction pathway. And similarly, for mesenchymal differentiation also, these pathways are very, very important. So that way they are linked. The mechanical stimuli, the mechanical environment has tremendous role in stem cells differentiation phase. That's why when I was talking about the media or the medium or stem cell niche that we are providing for its culturing, that reflects upon the its differentiation fate. So in which fate, in which direction the stem cell that we are culturing will be going on, will be going, right? So this is one type of a linear actuator. So you can think of like this kind of only a single direction motion is possible by this linear actuator. We can also attach a second motor at the perpendicular direction and then give it a bidirectional motion. So at the same time, a 1D as well as 2D motion can be given. Okay, so 1D and 2D stretching can be provided for mechanical stimulation. These are the really basic and simple design for mechanical stimuli, external mechanical stimuli providing. Right. Another mechanism is we have talked about this magnetostriction. The mechanism is called magnetostriction. What is that? That is not just the we have studied about the ferrogels, right? So what ferrogels does upon the magnetic stimulation or upon in the vicinity of a magnet, the ferrogel constricts. As when the magnet is not there, it releases. So this constriction and then release, this motion gives the cells that are entrapped in it its mechanical stimuli. So depending upon this phenomena, that is the magnetostriction, that is constriction upon the magnetic field induction, we can call it a magnetostriction. So this magnetostriction phenomena of ferrogels can also be taken for the stem cells differentiation. So magnetic field as well as this physical stretching. Third one is acoustic, that is ultrasonic. Not only just the electrical, magnetic as well as physical stretching, acoustic forces are also employed for providing external mechanical stimuli for cells. Like you can see, this whole dish, this is a 6-well plate, this whole dish is put upon a mono wavelength speaker. So a particular kind of frequency is generated and supplied to the culture dish, and the cells are cultured. Okay, the cells are seeded in this culture plate. You can see these rings are created, right? So this is the sound wave that is coming out, and depending upon this particular frequency range, we can change the particular frequency that we are treating the cells with. And you can they have studied about it that the stem cell differentiation property changes, their fate changes depending upon this kind of sound waves that are being generated or treated the cells. Okay, so this stretching as well as acoustic and magnetic actuation significantly impacts the cellular or particularly stem cells differentiation rate. Okay, so this is I have put this example just to show you this kind of bioreactor system is used to study this kind of behavior or mechanical stimuli, how to provide the mechanical stimuli, what are the changes that are occurring. So you can think about this way. So when we are providing a mechanical stretching, it is just not only just this motion of the cells. So we are constantly supplying the cells with a particular media. We are providing the media, we are supplementing with it. And there are some dynamic bioreactors also. So where there is a particular fix with this kind of lines of media. You can see these are the media bottles. So there are lines of media that are going in and going out, right? So this forces, this media going in and going out, this also generate different kind of forces to the cells. And depending upon this kind of mechanical stimuli, the stem cell property can also change. Here, this setup has a ultrasound detector. Okay, so not only just visual like optical imaging, we can also set these things like we can also tailor this kind of bioreactor systems and get ultrasound as well as this. Here also there is a camera setup also in the paper you can find. So this kind of smart intelligent systems are being developed to take into advantage of the technology that are available to get the information on the cells. Okay, and see how the differentiation patterns are changing, how stem cells are behaving upon various other stimuli. Okay, so not only just do not think about the particular growth factor can only have its effect. There are various other factors as like mechanical factors and as well as other like the properties of ECM that you have studied in the lecture. So this kind of factors are also impacting the stem cell differentiation property. Okay, right. Okay, right. Next question is in asymmetric cell division, two daughter cells are. It's a pretty straightforward question. In asymmetric cell division, the two daughter cells are identical, non-identical, or differentiated. Okay, I'm getting the response. Anyone? Right, right. So they are non-identical. Right. In asymmetric cell division, the two daughter cells are always non-identical. So one set is having like for the same cell differentiation, they are having the asymmetric cell division, right? Cell division, right? So one cell has the particular self-renewal property, the other is lineage specific. Right. So the asymmetric cell division, the two daughter cells will not be identical. So they are non-identical. Okay. Next, the question comes is fluorescence activated cell sorting facts we have studied uses what in order to isolate specific cells. Option is surface markers, density, size, and morphology. Right. Anyone? Anyone else? So these are surface markers, right? Yes. So the particular surface markers are immune markers like CD45 or CD24. This kind of uh, surface markers are being investigated upon to employ it in FACS-based cellular detection systems. So these are the high precision systems where depending upon the what are the surface markers that are created upon the cell's surface, this kind of cell sorting as well as cell identification can be performed. Don't think of FACS as like always it will have a cell sorting unit attached to it. Mainly, FACS gives us an idea about the what are different kind of cell surface markers that are being expressed and what kind of population of cells we are particularly dealing about. But when it is attached with a cell sorter unit, it comes as a different attachment. So FACS with cell sorting, fluorescence activated cell sorting, a cell sorter comes as a separate attachment, and when this attachment is installed, we can actually perform this cell sorting depending upon the particular cell surface marker. All right. So what is the technique that uses the cell density, size, and morphology of the cell to differentiate, to isolate the specific cells? What would be the technique? Cells density, size, and morphology. Which technique uses to isolate a particular cells? Anyone? Try. The option is there. It is density gradient centrifugation, right? That technique uses the particular cell density as well as size and morphology to not particularly morphology, but density and size. And morphology also has an impact uh, in the density gradient centrifugation. So depending upon if the morphology changes to a like if we have particular morphology of cell and a different morphology but of same density, then there would be a change in their level, very minute, but their level would be a little bit different. So we can also use that technique for isolating that particular cell. But yeah, the technique that we can use for this, like density dependent, size dependent cell isolation, is density gradient centrifugation. Right. Okay. Right. So the particular cell surface markers that we are used for FACS, density gradient centrifugation, sorry, density gradient centrifugation that we have studied, right? So various density is created either discontinuous or continuous, depending on discontinuous phase or continuous phase we have created, right? We have studied about it. So for blood cell isolation, we have done this is density gradient centrifugation. That what it uses. It uses various different kinds of cells densities, right? The density and cell size that we are having in our blood. Depending upon that, we are getting the separation. Cell density and size, which technique uses density gradient centrifugation. Right. Right. So the particular type of cell surface marker that we are investigating upon can be identified by various mechanisms. One of the very popular one is antibody based. So when the FACS is concerned, what we used to do is we tag the antibodies with a particular fluorophore. For example, there are various fluorophores like FITC or Alexafluor or site 633 is depending upon the emission spectra, emission peak, the fluorophore name changes. So it can be like site 564, site 663. Depending upon this peak position, the fluorophore name also changes. So very common and very commonly used fluorophore is FITC, FITC level antibody, and their conjugation kits also come in the market, and their conjugation step is really easy. So what it does is like particular cell surface marker specific antibody we have, we can generate the antibody and tag it with a particular fluorophore. And when we treat our cells with this kind of fluorophore, whatever the antibody that the fluorophore is that the cell surface marker that the antibody is binding with, so the fluorophore is also binding with the cells and it is tagging the cells with the particular kind of fluorophore. And depending upon that fluorophore intensity that we are getting in the FA in the FACS, we are then isolating or analyzing, or if there is a sorter, then we are sorting the particular kind of cell. That is the FACS technology. But this FACS and its yield is also very less. This fax technology uses a very big space. Like these are bulky machines. You can think of a half a room full of machine to have actually a particular kind of cell sorter, fax, nice quality cell sorter. If we install it, it would be like a half full of room. As well as there's a bulky machine and lots of reagents, media, etc. is required. So this venture is a really costly one. So what is an alternative? Microfluidic technology came in. Microfluidic technology, what it does, it is either of, it is of two types, but majorly microfluidic technology have certain advantages. That is a small sample volume, as well as the reagent consumption that occurs for the cell sorting is very low. It requires very less space, as well as it is a, so this all the things make it a sustainable venture, right? So microfluidic based system provides an alternative to the cell sorting technology for not only just for this stem cell or particular cell isolation. This microfluidic based approaches are now being commercialized for cancer stem cells, that is the circulating tumor cells or cancer stem cells identification also. Their morphology also changes. So microfluidic based cell sorting is by property or by principle can be of two types. One, it can operate by an active mode or the passive mode. So what passive mode is, it doesn't matter what kind of surface property or what kind of cell we are dealing with. It is for all the type of cells that we can think about. It employs cells, or it employs the cell's size, density, as well as those property that are property of the particular cell and doesn't matter the kind of cell we are dealing with, right? So there is no need for any kind of other treatment to the cell. Just the, let's say the, we have two kinds of cells. One is a big hepatocyte cell, and other one is a very small fibroblast cell, right? Or a blood platelet, or a very small cell. Not, let's not go to the example. Like a big cell and a small cell. Now, depending on the microfluidic channels orientation, we might be having a very kind of a mesh in the channel, or kind of a pillars in the channel length, or we can also have the channel constriction, microfluidic channel constriction. So depending upon this kind of change in the channel properties, we can sort, like the small cell will pass up and go to a separate reservoir, and the big cell that will be reserved, that will be retained, will can get into a different reserve, right? So you can imagine a particular microfluidic channel with two reserves and one inlet, and there are various channels that these things are going through, and there are pillars in the channel way. So depending upon the particular cells, if the small cell can pass through the pillar, then it is going to another reservoir, and if it can't, then it goes to a second reservoir. So this kind of mechanism of cell sorting is, we can call it like a passive mode that doesn't require us to treat the cells with any kind of external force or external reagent or anything. But there is another mode that also employs this microfluidic based system, that is this active mode, where either electrical, magnetic, or optical based cell sorting can be performed. So see, the microfluidic platform can be fabricated with very transparent material, as well as we can fabricate it with various kind of probes. And using those kind of probes, as well, like electrical probes, as well as this optical platform, we can treat our cells with various kind of fluorophores, as well as like same like aptamer tagged fluorophore or antibody tagged fluorophore. We can treat our cells with, and then see, visually see in the channel only, and then sort it out with the help of various other mechanisms. Right. So this kind of sorting, where we employ a various reagent and external force, we call it as an active mode. So depending upon this, either passive mode or active mode, cell sorting can be performed with very low sample volume. So we do not need to, we can also we can also get a live image from a sample collected from a patient, and we can just let the sample run through this microfluidic channel and sort the cells out depending upon its properties. What the particular kind of cell we need, we can particularly change the microfluidic channels property, and we can sort the cells out very easily. We don't need this kind of huge bulky machines, right? So this is an alternative technology that is being investigated upon recently, that is microfluidic based cell sorting. Okay. So this is an alternative to fax that is coming up, and it is bridging the gap of various labs that doesn't have all that amount of money to afford a fax system. Well, not only just labs, very institutes might not have that kind of money to afford that kind of a fax system. So for them, this kind of microfluidic based cell sorting system can provide a really advantageous platform to have it in a lab and to analyze our samples. As well as these platforms can be used for various clinical purposes only. Doctors, they can just very easily read the sample and sort the cells and do their analysis. Right. These easy platforms are currently being investigated. Right. So we have talked about aptamers. Like aptamers, we have talked about. Do you know what are aptamers? Like what are aptamers based sensing? What is this aptamers? We have known antibodies, right? Right. Yeah. It's either can be a DNA based aptamers or RNA based aptamers, but what it employs is the secondary structure that is being produced by this kind of nucleotides or strands of nucleotides. The secondary structure interacts with the particular surface marker. It can be a carbohydrate, it can be a protein, or whatever the cell markers. Even the bacteria, even whole bacteria can be sensed by this aptamers. Okay, the bacterial aptamer sensors there. So this kind of whatever the particular site that needs to be that sensed, the aptamers, the particular kind of oligo can have, or particular kind of nucleotide sequence can have this secondary structure, and depending upon that, and it binds to that structure. Depending upon the particular secondary structure it attains, the binding event occurs. And if we tag this kind of secondary structured nucleotide sequence with a kind of fluorophore, like again FITC labeled aptamer, then we can perform this kind of cell sorting. So aptamer, that way it works. And for finding the particular kind of aptamer, what is the technique that is used? Just for like a brief information, it is called SELEX. Okay, just get acquainted with the term. SELEX is the SELEX is the technique where we can find the particular kind of aptamer that is interacting with the site of our interest. Right? This is just a brief aptamer. As we are talking about antibodies and aptamers, I just thought like we should discuss a little bit. Okay. All right. So this is my part today for the discussion of the questions that I wanted to talk about. This week I didn't find that much of a question from you. So if you have any question, please do ask. I am opening the chat section. Is okay. It is open. Yeah. You can write the questions down if you have any questions. So the content of this week was the cells, whatever the cell isolation, stem cells particularly, their differentiation pattern, and okay, right, right, okay. And I also had some these hands, right? So I am turning on your mics, right? So okay, can you get only the presentation slide instead of the transcript? Yeah. So the issue is the NPTEL policy probably does not support the slide sharing currently, and we will be discussing with the NPTEL authority as well as also Vignesh regarding if it is possible to share the slides. But yeah, for now, for your study, you can use that text transcript and make use of it. But yeah, I will discuss this issue with. So, but the thing is, the text transcript would be helpful to give you a brief overview of what was talked about uh, for that particular slide, right? So you can think of like if it can help you to supplement whatever the slide content that is there. Okay. Regarding week 4 assignment, you have some questions. You can unmute your mic or you can write those down in the chat section. Yeah, that would be better. This is based on conclusion of basic metric. I think there isn't any substance which is called basic fuel. So I wait for a single question. Statement shows that I can't understand how statement shows right. Right. Okay. If you have like this issue, yeah, this is urine to fluorescent urine transfer is the cause of this fluorescent detection. If you think that this option should be correct, I think you should write this question to the discussion forum. And, we will see what can be done. Okay, don't worry. It will be resolved. Don't worry. You just write this to the discussion forum. We will be resolved. Right. In the middle of the class, Saloni was raising hand probably. Saloni, do you have any question or anyone else? Right. Oh, okay. Hmm. Okay. Cool. So anyone else? Yeah, discussion forum. Mushami, discussion forum. You write all those questions. This question along with the query, like the particular question you are talking about, and the correct answer that is given. Keep it and you upload it with the question that why this is not showing as a correct option. Okay, we can get back from there, right? Okay. All right. So this week content, the whole thing was to see how the cells and what particularly stem cells as well as the hematopoietic stem cells, how the stem cells differentiation and behavior changes with the various factors and how it is important for our tissue engineering purposes. Why? Because not only just this cells are. Why did we study these cells and the behavior of cells? Because this tissue engineering platforms that we have studied previous weeks, we can employ this tissue engineering techniques with this stem cells properties as well as stem cells knowledge with this tissue engineering knowledge, and then employ this both as a novel platforms to address a particular situation. Okay, so both this knowledge is that goes hand in hand, and we need to develop various platforms that employ both these parameters right now, because smart systems, smart hydrogel based or smart cell based systems are now being researched upon or being employed much more greatly, because not we just providing the cell with a particular solid construct and expecting the cells will behave depending upon the properties of the construct. It is a bit of overstage. If we are supplementing the particular type of cell along with it, that won't create any kind of immune response. That would be a better platform for our tissue engineering purposes, right? uh, Anyone else? What happened in case of scaffold? What kind of scaffold you are talking about? From previous problem solving class, I know NMR is also to use to measure the porosity of my sample. Then what happened in case of scaffold? Like for the particular porosity determination, NMR can be employed. So not only just like a particle or any kind of material, it can be employed to any kind of material, like a spora scaffold also. It can measure the porosity of the scaffold also, right? This is that is what you are asking, right? So it is possible to measure the porosity with the help of NMR to the scaffolds. Not only just the particles, what are the particle surface area, it can be employed to measure the scaffold's porosity also, along with PET and along with the porosimetry and everything. Right. Did that answer your question? Question? All right. So, anyone else? Do you have any other question? Okay. Uh, there is a question. Okay. Right. This question, which of the technique we use? Choose anyone. Right. Right. See. Right. So you can write this question also in the discussion forum. Please do write this question so that we can get back. Because we have to discuss with IPTTAs and we will be getting back to you. Don't worry, these questions will be addressed. If you think that if you, whatever, if you have any uh, confusion regarding the question and the answer in the assignment also, please do write there. But not before the next week content or assignment submission deadline is finished. So just make sure that the deadline is passed, then you write it. Okay. Don't worry, it will be answered. Okay. All right. So yeah, please do write all the confusions and all the questions to the discussion forum so that we can collectively get a decision and we can address the problems. Okay. And go through the content and discuss amongst yourself and discuss all the materials that are being provided and thoroughly. And I have also provided the materials that I talk about in the slides, and you can get to the slides with the link that have been provided. So go through those links and study about it and write the whatever the problems that you are facing to the discussion forum. Okay, as much as you want, you use the discussion forum, and we will be getting back to you. Okay. Thank you for joining the session as well, and I hope that we see you the next week. Okay, thank you, thank you. You can leave the session.